Welcome to our microlearning channel. Today, we will focus on the plants and more specifically the essentials of plant development. Auxin is a vital plant hormone that regulates many important developmental processes. It is crucial for stem elongation, root growth, and the formation of lateral roots and buds. This hormone helps coordinate the plant's growth and development across different tissues and organs. Understanding auxin biology is key to understanding how plants grow and respond to their environment. The image on this slide shows a young seedling emerging from the soil, illustrating auxin's role in early plant development. Auxin is a crucial plant hormone that regulates various aspects of plant growth and development. It primarily promotes cell elongation, leading to stem growth and root development. Auxin exhibits polar transport, moving primarily from the plant's shoot tip to the base, affecting plant growth and development. This hormone plays a vital role in the overall growth and development of plants. Auxin is a critical plant hormone that regulates many aspects of plant growth and development. The primary pathway for auxin biosynthesis in most plants is the tryptophan pathway. This converts the amino acid tryptophan into indole 3-acetic acid IAA, the main form of auxin. There are also alternative pathways, like the IAM pathway and others, that contribute to auxin production. Auxin biosynthesis is tightly regulated by developmental cues and environmental factors. This ensures the precise spatial and temporal distribution of auxin, which is crucial for proper plant growth. Understanding the different auxin biosynthesis pathways and their regulation is an important area of plant biology research. Auxin is a key plant hormone that plays a crucial role in plant growth and development. Auxin transport is a tightly regulated process that involves several mechanisms. Polar auxin transport moves auxin unidirectionally from shoot tips to roots, driven by specialized transporters. Active transport requires energy to move auxin against concentration gradients, using protein pumps. Auxin can also move passively through cell membranes by diffusion, following concentration gradients. Pin proteins are a family of auxin efflux carriers that facilitate the movement of auxin out of cells, enabling polar transport. Auxin influx carriers help bring auxin into cells, contributing to the overall transport process. Understanding these auxin transport mechanisms is crucial for regulating plant growth and development. Auxin is a key plant hormone that regulates many important processes in plant growth and development. The signaling pathways triggered by auxin are complex, involving several steps from perception to changes in gene expression. First, auxin is perceived by specific receptor proteins, which initiates a downstream signaling cascade. This signaling cascade involves the activation of various proteins like kinases and phosphatases. These signaling components ultimately lead to changes in gene expression within the plant cells. By altering gene regulation, auxin can drive diverse cellular responses that shape plant growth, development, and adaptation. Understanding these auxin signaling pathways is crucial for understanding how plants respond to and utilize this important phytohormone. Auxin is a key plant hormone that plays a crucial role in various aspects of plant growth and development. Auxin promotes cell elongation by stimulating the production of enzymes that loosen the cell walls, allowing the cells to expand. Auxin from the apical bud, the tip of the plant, suppresses the growth of lateral buds, ensuring the plant grows vertically and maintains its dominant growth pattern. Auxin is essential for root formation and elongation, promoting the growth and development of root tips, which are crucial for nutrient and water uptake. Auxin also promotes shoot development, influencing the formation of new leaves and branches, contributing to the overall growth and architecture of the plant. Understanding the role of auxin in these key processes is crucial for understanding plant growth and development and has important implications for agriculture and horticulture. The image on this slide shows a close-up view of a plant cell undergoing elongation, with a focus on the cell wall, cytoplasm, and organelles. Auxin is a plant hormone that plays a key role in promoting cell elongation. Auxin increases the plasticity of the cell wall, allowing the cells to expand and lengthen. Auxin stimulates the activity of proton pumps in the cell membrane, which acidifies the cell wall and weakens its structure. Auxin also regulates the expression of genes involved in cell wall modification and expansion, promoting the synthesis of enzymes that break down cell wall components. These processes all contribute to the overall elongation and growth of plant cells. Auxin, the plant hormone produced by the apical bud, plays a crucial role in maintaining apical dominance. The high auxin levels near the apex suppress the growth of lateral buds, directing the plant's energy towards vertical growth and maximizing its access to sunlight. Auxin is transported downwards through the stem, creating a gradient with high concentrations at the top and low concentrations in the lateral buds. Removing the apical bud disrupts this auxin transport, allowing the lateral buds to develop and the plant to branch out, increasing its overall size and leaf area. Understanding the mechanisms of auxin-driven apical dominance is valuable for optimizing plant architecture in agricultural crops, improving yield and resource utilization. Auxin is a key plant hormone that plays a crucial role in root development. Auxin promotes the elongation of the main root by stimulating cell division and elongation in the root apical meristem. This allows the root to grow deeper into the soil, improving the plant's anchorage and ability to absorb nutrients and water. Auxin also creates concentration gradients within the root that direct its growth towards gravity, ensuring the root system develops in the optimal direction. Auxin triggers the formation of lateral roots, which branch out from the main root to increase the overall surface area for nutrient and water absorption. The precise control of auxin concentration gradients within the root regulates where these lateral roots will form and how they will be positioned. The apical bud of the plant produces auxin, a key plant hormone. This auxin suppresses the growth of lateral buds, promoting the vertical growth of the main shoot. This phenomenon is known as apical dominance, and it helps the plant maintain a central, upright growth habit. Auxin is transported downwards through the shoot in a polar fashion. This polar auxin transport influences the distribution of resources and regulates the development of new organs like leaves. Auxin also promotes cell elongation in the shoot, contributing to stem growth and the emergence of new leaves. Additionally, auxin can influence the formation and growth of lateral buds, shaping the overall architecture and branching pattern of the shoot. Auxin is a key plant hormone that influences various growth responses, including phototropism, gravitropism, and thigmotropism. In phototropism, auxin accumulates on the shaded side of the stem, causing increased cell elongation and bending of the stem towards the light source. Gravitropism is the growth response of plants to gravity. When a plant is placed horizontally, auxin accumulates on the lower side of the stem, promoting cell elongation and causing the stem to grow upwards. Thigmotropism is the growth response to touch or contact. When a plant touches a solid object, the distribution of auxin is altered, leading to differential growth and bending of the plant towards the object. Understanding the role of auxin in these tropisms is important for understanding plant growth and development, as well as potential applications in agriculture and horticulture. Auxin is a key plant hormone that plays a crucial role in various aspects of flower development. Auxin promotes the formation of floral buds by regulating the transition from vegetative to reproductive growth. This ensures the plant can produce flowers. Auxin also controls the size and shape of floral organs like petals, sepals, stamens, and pistils. This ensures proper development of the flower. Auxin plays a role in pollen grain development and maturation, which is essential for successful fertilization and seed production. Auxin influences the timing of flowering by regulating the expression of genes involved in the transition to flowering. This allows the plant to flower at the optimal time. Overall, auxin is a critical hormone that coordinates various aspects of flower development to ensure the plant can reproduce successfully. Auxin is a crucial plant hormone that plays a vital role in fruit development and growth. It influences the size and shape of fruits by regulating cell division and expansion. As fruits ripen, auxin levels decline, triggering the production of ethylene, which leads to color changes, softening, and flavor development. Auxin also regulates fruit abscission, the process by which fruits detach from the plant, ensuring efficient seed dispersal. Auxin promotes the development of flowers into fruits, ensuring successful seed production, and also influences the overall size and yield of fruits. Auxin plays a complex role in leaf senescence, delaying it in young leaves but promoting it in older ones. Auxin regulates the expression of genes involved in senescence, influencing the breakdown of chlorophyll and other cellular components. Auxin promotes the mobilization of nutrients from senescing leaves to younger, developing tissues, contributing to plant resource allocation. Environmental factors like light, temperature, and nutrient availability influence auxin levels, which in turn impact senescence processes. Auxin is a key plant hormone that helps plants respond to various environmental stresses like drought, salinity, and heavy metals. It does this by regulating processes like stomatal closure, root growth, and the accumulation of protective compounds, all of which enhance the plant's tolerance and ability to survive these stressful conditions. Auxin also plays an important role in the plant's defense against biotic stresses like pathogens and herbivores. It contributes to the production of defense hormones, the activation of defense genes, and the formation of physical barriers like callose deposits, all of which help the plant ward off these threats. So, in summary, auxin is a versatile hormone that allows plants to adapt and respond to both abiotic and biotic stresses, helping them survive and thrive in challenging environments. Auxin is a key plant hormone that mediates various growth responses to environmental stimuli. Hydrotropism. Auxin helps roots grow towards water sources, ensuring plants have access to this essential resource. Phototropism. Auxin controls the bending of stems towards light, optimizing photosynthesis for growth. Gravitropism. Auxin regulates the downward growth of roots and upward growth of shoots, maintaining proper orientation. Thigmotropism. Auxin plays a role in touch responses, enabling plants to climb and anchor themselves to physical supports. Auxin plays a crucial role in establishing beneficial relationships between plants and microbes, such as nitrogen-fixing bacteria and mycorrhizal fungi. Microbes can influence auxin production and transport, affecting plant growth, development, and stress responses. Auxin acts as a signaling molecule, mediating communication between plants and microbes, influencing nutrient uptake and pathogen defense. Auxin is involved in the formation of root nodules, where nitrogen-fixing bacteria reside, and in the development of mycorrhizal associations. Auxin is a critical plant hormone that plays a central role in regulating growth and development across the plant kingdom. Farmers can leverage auxin's effects to optimize crop yields, for example, using synthetic auxins to promote fruit set and development, ensuring proper size and quality. Auxin-based herbicides selectively target weeds, which tend to be more sensitive to auxin, allowing farmers to control unwanted plants. Researchers are also exploring ways to engineer auxin-related genes to improve stress tolerance and disease resistance in crops through biotechnology. Overall, understanding and harnessing the power of auxin is crucial for advancing modern, sustainable agriculture. Auxin is a critical plant hormone that regulates many aspects of growth and development. We can leverage our understanding of auxin to enable powerful biotechnology applications. CRISPR-Cas9 can be used to edit genes involved in auxin biosynthesis, transport, and signaling. Engineered auxin-responsive promoters can control gene expression to enhance plant growth and development. Auxin biosensors allow for accurate, real-time monitoring of auxin levels in plants. Harnessing auxin-related technologies can improve crop yield, stress tolerance, and nutrient utilization. Auxin is a key plant hormone that regulates many aspects of growth and development. Genetic engineering allows us to manipulate auxin levels and signaling pathways in plants. By modifying genes involved in auxin biosynthesis, transport, and signaling, we can fine-tune auxin's effects. This can lead to enhanced crop yields, improved stress tolerance, and other desirable traits. For example, altering auxin transport genes can control the direction of growth, leading to more branching or deeper root systems. These targeted modifications could result in crops with higher fruit production, better nutrient uptake, or drought resistance. The ability to harness auxin's power through genetic engineering is a promising avenue for improving agricultural productivity. Auxin, a key plant hormone, can influence epigenetic mechanisms like DNA methylation and histone modifications. These epigenetic changes can alter gene expression patterns in plant cells. Auxin-mediated chromatin remodeling can affect the accessibility of genes to transcription factors, impacting gene expression. Epigenetic modifications induced by auxin can have long-lasting effects on plant development and responses to environmental cues. Auxin-influenced epigenetic changes can help plants adapt to various environmental stresses, contributing to their phenotypic plasticity. Auxin is a critical plant hormone that regulates many aspects of plant growth and development. Systems biology approaches help us understand how auxin interacts with other hormones, genes, and environmental factors to coordinate these complex processes. Integrated network models can capture the intricate web of interactions between auxin and other signaling pathways. Computational modeling allows us to simulate auxin dynamics and predict plant responses to different conditions. High-throughput omics data provides a wealth of information about auxin-regulated genes and proteins. Network analysis techniques can identify the key regulatory nodes and interactions in auxin signaling networks. Understanding auxin signaling at a systems level is crucial for engineering plants with desired traits. Computational models are powerful tools for studying the complex dynamics of the plant hormone auxin. These models can simulate the transport, signaling, and responses of auxin within plants. This provides valuable insights into how auxin drives plant growth and development. Computational modeling can also predict how plants will respond to environmental changes or genetic modifications. This aids in the development of improved crops and plant-based products that are more resilient and productive. By optimizing our understanding of auxin-mediated processes, computational modeling helps improve agricultural practices, leading to increased crop yields, better disease resistance, and more sustainable plant-based solutions. Microscopy techniques like fluorescence, immunofluorescence, and confocal microscopy allow us to visualize the distribution of auxin and how plant cells respond to it. Biochemical assays help us measure auxin levels, enzyme activities, and signaling components in plant extracts. This quantifies auxin and helps us understand its metabolism and signaling pathways. Genetic approaches such as mutant analysis, gene expression profiling, and transgenic techniques identify and characterize the genes and functions involved in auxin-related plant development and responses. Computational modeling simulates and predicts the behavior of auxin gradients and signaling pathways, providing valuable insights into the complex interactions of auxin in plant biology. The history of auxin research dates back to the late 19th century, when scientists observed plant growth responses to light and gravity. Charles Darwin and his son Francis conducted experiments showing that a substance in the tip of coleoptils was responsible for phototropism. Later, Boyes and Jensen's experiments confirmed the existence of a diffusible growth-promoting substance, which was later identified as auxin. In the 1930s, auxin was isolated and chemically identified as indole 3-acetic acid IAA, a significant milestone in plant hormone research. Current research focuses on the molecular mechanisms of auxin synthesis, transport, signaling, and its complex role in plant development and responses to environmental cues. Auxin is a crucial plant hormone that regulates a wide range of developmental processes, from growth and patterning to stress responses. While we have a good understanding of auxin signaling and its immediate effects, there are still many unanswered questions about its complex mechanisms and interactions with other hormones. Delving deeper into auxin's signaling pathways and its roles in various developmental stages will be an important area of future research. Harnessing the power of auxin could lead to significant advancements in crop improvement, such as enhanced yields, disease resistance, and adaptation to climate change. Integrating systems biology approaches, including computational modeling, high-throughput screening, and genetic analyses, will provide a more comprehensive understanding of auxin action. Exploring the epigenetic mechanisms underlying auxin-mediated gene regulation. Auxin is a crucial plant hormone that regulates growth and development throughout a plant's life cycle, from embryo formation to senescence. Auxin mediates the plant's interactions with its environment, influencing responses to light, gravity, and various stresses, as well as interactions with beneficial microbes. Ongoing research on auxin will continue to advance our understanding of plant biology, leading to improvements in agricultural practices and biotechnology applications. The key takeaways are the central role of auxin in plant growth, development, and environmental interactions, as well as the importance of continued research in this area.